Hi everyone, have a good day. So this week the topic is topic 6, expression of biological information, which is a critical topic in your PSPM. Okay, let us see question number 1. Explain the enzyme involved in DNA replication. So what do you see here is a diagram, the position of the each enzyme and the function involved. Okay, it's very important for you all to know the position of the enzyme, because sometime in PSPM they might give you structural questions to identify the enzyme involved in the specific position. So you can see here there are total 7 enzyme. There are total 7 enzyme involved in a DNA replication. Okay, which is number 1 is a helicase. Number 2, what is number 2? Here, over here is DNA polymerase 3. Okay, right. Then where else do you see enzymes? Okay, we have primase, and we have DNA polymerase 1, we have DNA ligase, and we can't see the topoisomerase. Actually it's at the back here, topoisomerase. Okay, alright, so let us see the question. Okay, the answer. So they ask you to explain the enzyme involved in DNA replication. So when you mention one enzyme, you are eligible to get 1 mark. In the explanation, which is the function, you will get another mark. So total we have 7 enzyme, 7 times 2, 14 marks. Okay, you can write more to maximize your marks. Okay, right. So enzyme, first enzyme helicase, unwind the double helix DNA at replication fork by breaking the hydrogen bond between both DNA strand. Must write completely. Second one, single strand binding protein SSBP, but do not write abbreviation, write in complete term. Okay, bind to the unpaired DNA strand, prevent them from repairing or rejoining. So you have to write hyphen, because if you didn't write hyphen, become the word repairing, which is give different meaning. Okay, so the safe side you can write the word from rejoining. Okay. Right. Next is topoisomerase, help relieve the strain of overwinding ahead of replication fork, or help relieve strain by breaking, swiveling and rejoining DNA strand, or simple you can write prevent supercoiling. You already got 1 mark here and you have 1 mark here. Okay, so any simple sentence. Alright, next, primase synthesis, must use the word synthesize. RNA primer by adding complementary, must mention RNA nucleotide to the DNA template. So enzyme 1, function 1. DNA polymerase 1, remove RNA primer and replace with the complementary DNA nucleotide. Okay, so function, the name of enzyme and the function. Okay, DNA polymerase 3 synthesize new DNA strand by adding complementary DNA nucleotide to the 3' end of RNA primer, or you can write growing DNA strand, must write. Okay, the last, DNA ligase joins the Okazaki fragment. Okazaki, the O must be capital because it's the name of the scientist actually. Okay, into a continuous strand by forming phosphodiester bond. Okay, so you have to write completely in order to get 2 marks each of the enzyme. Okay, let us go to the next slide. Okay, overview about the topic. So this now was DNA replication. Okay, so next process, there are 3 major processes in this topic. The first one is DNA replication. Second one is transcription, where DNA is transcribed into mRNA. The last one is translation, where RNA is translated into protein. Okay, so there are sequence of steps involved. You all can read. Even it's the same thing from the note. So I just give you a simple diagram on the both process. Okay, if you see clearly, both process involve 3 stages: initiation, elongation and termination. You may read in detail in your notes, refer your notes. Okay, ask your friends to refer notes, bring their notes when doing the discussion. Okay, so same thing in translation, also 3 stages, but translation, if you read, there are 3 major step cycle during elongation. So bit detail. There are codon recognition, peptide bond formation and translocation. Okay, so let us move to the question first. Okay, so the first question, what is process Q? This is second question. Okay, figure 1 shows the process of protein synthesis in eukaryote. Okay, identify the process Q and its importance. Okay, so what is process Q? mRNA processing, RNA processing, splicing process. You cannot write transcription because this is very clear showing pre-mRNA processed into mRNA and the process Q is here. Okay, so transcription totally wrong answer. Okay, if it's a transcription, they only shows the diagram of the mRNA, but if you see this diagram is showing pre-mRNA processed into mRNA. So the critical process here is RNA processing. Okay, so another one is important. There's a two question here: to produce mature functional mRNA or to remove intron or non-coding sequence. Okay, so if you refer back to the earlier slide, you can see what is RNA processing. Okay, so it involves two step: alteration of the mRNA strand and split genes and RNA splicing. Okay, so what is the function? Why it need to undergo RNA processing? Okay, so what is the function of the 5' cap and poly A tail? Okay, so to promote the export of mRNA from the nucleus, help to protect mRNA from degradation, help ribosome to attach to 5' end of the mRNA in cytoplasm. Okay, so all these are from your notes. Okay, next it will undergo splicing to remove intron, which is the non-coding, and join the exon to create an mRNA molecule with the continuous coding, which is conducted by spliceosome. Okay, so you have to read all these. Okay, next. I want to check whether it's recording or not. It is recording. Okay, so let us move to question number 2. Okay, term referred to the molecule A. So what is molecule A? So over here always refer. So this is tRNA. Okay, this is tRNA. So reflect the 333 bases. Practice yourself to underline the keywords when you read. Okay, so refer to the anticodon. Okay, next question, question 3. Round the produced by process R base sequence is on mRNA. So I'm highlighting. Make sure it is mRNA. Okay, so if you refer to the earlier slide. Okay, earlier slide. Our genetic code is based on the mRNA. So you no need to memorize. You no need to memorize the genetic code. It will be given like this during your PSPM. Okay. So you have to make sure it is given is mRNA, and you can do the direct translation. Okay. If it's not on mRNA, it is given DNA. Okay. If it's given DNA, then you have to transcribe into mRNA. If it's given is anticodon, so you have to get the co-codon because our genetic code based on the co-codon. So remind your friends and make sure it is mRNA. Okay, right. So we see the first one CGU. So this one direct translation because it's given is mRNA. You no need to do transcription. Okay. Right, so CGU for arginine, okay, GUG, so it's read by the 3 bases, GUG, okay, and accordingly, next is methionine and last is CAC for histidine. Okay, if you see here, AUG is not given, but you should know AUG is the start codon. Okay, so start codon, you should know start codon is methionine. Even though it's not given here, at least you remember or remind yourself. Make sure you remember. Make sure you memorize. Okay, AUG is a start codon is methionine. So do not omit to write it because it's not in the list. No, it's not like that. You must write it accordingly. Okay, next is differentiate the process P and process R. Okay, so what is given here? Process P. Where's process P? Over here. So P is, we can see that DNA is transcribed into pre-mRNA. It's not a DNA replication because clearly we can see it transcribed into mRNA. And process R is a very clear, so outside the nucleus, it is the cytoplasm, so it is a translation. So you no need to mention what is process P, what is process R, no need to mention, just use the given letter. But if you want to mention, you have to mention early, you have to introduce process P is transcription while process R is translation, but marks is not given. Then after that you can replace, transcription takes place in the nucleus, so after that you can replace, but you are not advised. Okay, so use the given word. Okay, so compare A to A. Okay, and then use the connector while, you can use the word but, whereas. Okay, so total 3 marks, you must write 3 differences, but here got 4. It's good if you write extra. So process P takes place in the nucleus while process R take place in the cytoplasm. Process P use DNA strand as a template, must write completely, while process R use mRNA as a template. Process P produce single strand mRNA while process R produce protein or polypeptide chain, either one. Okay. Process P involve enzyme RNA polymerase 2, must write in Roman, while process R involve enzyme amino acid tRNA synthetase. The spelling must be correct. Another one is peptidyl transferase, must write in correct spelling. Okay, next question, direction. So direction always 5' to 3'. During move along mRNA. So mRNA is towards that side. So it is 5' to 3'. Okay, the bond between, this one also in chapter 1. The bond between amino acid is the peptide bond. Okay, so make sure you read this process in detail. So next we see mechanism of the lag operon. So you may read your notes, what happened to this operon. Is it switch off, is it switch on when lactose is absent? So very easy concept. When it's absent, of course it is switch off. If it's a present, it will be switch on. That's all. Okay, so easy concept, you may read your notes. We go to the question. What's meant by operon? So this is a definition question. You have to write what is written in your notes. Cannot create your own definition or write your own sentence. Okay, so it is 2 marks. You have to completely write. A series of structural genes expressed as a group and controlled by the single promoter. Okay, what happened to C? So C, if you are not sure what is C, you no need to write as a repressor. You just mention as a C. And C detach from B. You are not sure, don't write. If you are sure, you can write what is C and what is B. Okay, this is only 1 mark. What happened? That's all, 1 mark. Okay, give reason for your answer. Why is detached? Because the milk contain lactose and glucose. Okay, but if you see here, the lactose is not metabolized. That means the lag operon actually is switched on, but it's not fully functional because the bacteria prefer to use the glucose as a main energy source. Okay, so once the glucose is completely used, then only it started to use the lactose. So where the lag operon is fully switched on. Since it's only 2 marks, you can give any 2 points. Okay, function of the product encoded by gene Y. You can see there's a 3 structural gene. Each one of these have their own function. So you have to read. So this one asking gene Y. So gene Y is permease. You no need to mention the name if you are not sure, but you know the function. Okay, so scared it will be wrong answer. Okay, but you're supposed to know what is gene Y. It is a permease. Okay, facilitate the transport, or you also can say uptake. You also can give this word. Okay, facilitate the transport or uptake of lactose in E. coli or increase the permeability of cell membrane to lactose. So only 1 mark. So we have 3. Make sure you all read all 3 function, all 3 of the structural gene. Okay, so I think that's all for today. All the best. Make sure you teach your team members carefully, check their answers also. Have a nice day. Bye.